we have three learning objectives today for our notes. First of all, we want to identify the chemical structures of DNA, and then actually two that we're going to start with. Then we are going to look at the clues that help scientists determine the structure of DNA. Specifically, we are going to take a look at Chargaff's rule specifically. So at this point, we know that there was the Griffith experiment, the Avery experiment, and the Hershey Chase experiment. And at this point, we know that DNA is the molecule of genetic inheritance. But now, to understand how DNA does its three jobs, and remember the three jobs of DNA are that it has to store information, genetic information, it must copy genetic information, and it has to be able to make that genetic information work and or be transmitted to the next generation. So in order to understand how DNA does its job, we have to understand the structure of DNA, because before you know how something works or why something is able to do its job, you have to know how it actually works. Okay, so sliding down to where it says nucleotide structure. Oops, I need to kind of get over here. There we go. Oops, went too far. All right, so the DNA is what we call a nucleic acid. Um, And remember, nucleic acid is one of the four main macromolecules. So, way back in like September, October, we talked about the four macromolecules, those four macromolecules that are present in every living thing are going to be carbohydrates, lipids, proteins, and nucleic acids. So, DNA is just one of our nucleic acids. Um, and again, if you remember about the macromolecules, these are long, long strands or chains of things we call polymers. Polymers mean that they are held, like there's a bunch of small little parts held together, and they are held together by covalent bonds. Um, so when we take a look at nucleic acids, we also have to take a look at their monomer. The monomer of a nucleic acid is what we call a nucleotide. All right, so the picture that you are seeing there on the side that is labeled 1, 2, and 3, that is a nucleotide. Number 1 is what we call the phosphate group. I don't know why, so please don't ask me, but in every textbook I have ever seen, the phosphate group is always symbolized by like a circular structure. Labeled as number 2 in your notes, we have the deoxyribose sugar. Deoxyribose is literally just the name of the sugar, kind of like, you have a name, I have a name, that sugar has a name, and that sugar is deoxyribose. And then number 3 in your label, in your diagram, that is the nitrogen base. So again, every time you look at a nucleotide, and again, the nucleotide is the building block of DNA, actually of any nucleic acid, DNA or RNA. The circular thing is always referred to as the phosphate group. The pentagon, the five-sided object in the middle there, is always going to be the sugar. And then the shape of the base can sometimes change, but it is always a nitrogen base. Now, if this was RNA, instead of having deoxyribose as the sugar, we would simply have ribose as the sugar. So, one of the big differences between DNA and RNA, deoxyribose nucleic acid and ribose nucleic acid, is literally just the sugar that is at the center of that nucleotide. They have different sugars, so they're going to have different chemical compounds or different chemical components, I should say. All right, so here is our structure of DNA. DNA is made up, actually, all nucleic acids. I keep saying DNA, but I really should be talking about nucleic acids in general. All DNA or all RNA has a structure that looks something like this. Now, Now you'll notice the labels A, G, C, and T. Um, that component, one phosphate, one sugar, one base, is what we refer to as one nucleotide. There are covalent bonds between each nucleotide that holds the nucleotides together. All right, so now we are going to go ahead and continue looking at the notes. If I look at the figure over here to the side, it only has 4 uh, nucleotides shown. Keep in mind that an average strand of DNA literally has millions of nucleotides. Millions. Like our little diagram in our notes shows 4. It shows an A, a G, a C, and a T on those 4 nucleotides. Every strand of DNA has millions. Now, what makes DNA different? Well, DNA has 4 different types of nucleotides because it has 4 different nitrogen bases. So every DNA molecule is made of nucleotides, but there's 4 types of nucleotides. The A is what we call adenine. G is guanine. C, oops, went too far. C is cytosine, and T is thiamine. Now, keep in mind, these nucleotides are held together by the covalent bond. Remember, covalent is when we are sharing electrons. There is a covalent bond between the sugar of one nucleotide and the phosphate of the next nucleotide. Now, these 4 different nucleotides act like the 26 letters in the alphabet. So I want to think about it. Like in English, we have 26 letters in our alphabet. However, if you pick up an Oxford English dictionary, the last time I Googled it, there's something like 5 million different English words, like totally different. Not like when we say like run, running, runner, like not derivatives, but actual 5 million different words in the English language. And we come up with those 5 million different words using 26 letters. Again, using think of it as a 4-letter alphabet, we can come up with literally millions of combinations of genetic code. So that's why, even though we have 4 nitrogen bases, how you put them together is what makes a different genetic code. So somebody might be like in the picture, we had AGCT for a gene. Maybe yours is AGTC, and then somebody else's might be GACT. So how you put these nitrogen bases together is what makes different combinations of genetic code, which is what then creates different genes different traits on different genes. So this is why maybe somebody has blue eyes, somebody has brown eyes. We have different combinations of these nitrogen bases. All right, so when we're looking at solving the structure of DNA, ladies and gentlemen, and like I said, we are going to just stop with Chargaff today. There's no need to go any further. So one of the puzzling facts about DNA, what was curious, is the relationship between the nucleotides. Chargaff discovered, excuse me, that the percentages of adenine and thymine were always equal in any sample of DNA. So he did fruit, he did vegetables, he did animal tissue, he did trees, everything he could get a hold of. He literally looked at the DNA and he found that the percentages of adenine and thymine were always equal in any sample of DNA. And this is also true for the other nucleotides as well. The amount of guanine was always equal to the amount of cytosine. Okay, so given this, that is Chargaff's rule that adenine and thymine are always present in equal amounts, guanine and cytosine are always present in equal amounts. Okay, so given this, if I had 30% adenine, how much thiamine would I have? Anybody? If adenine and thymine are present in equal amounts and I have 30% adenine, how much thiamine am I going to have?
30.
30% as well, because again, they have to be equal. All right? So there's my 30. All right, so if I have 30 and 30, how much of my 100% is left? So 30 plus 30 is what?
60.
60. So if there's 60% that I know of already, how much of my 100% is left over?
40.
40. And here's the thing, guys. I know I have to split that 40 between guanine and cytosine because there's only 4 nitrogen bases. So as long as you know one, you know the rest of them. Does that make sense? Yes, no, maybe, questions?
Yeah.
All right.
Yes.
Awesome. Awesome, 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 awesome. All right. So, ladies and gentlemen, let's try it again. Oh, let me clear my board. This is somewhat similar to what we'll be doing today. Okay? So again, this is actual data from Chargaff's experiment. Now you have to understand in any time that we do an experiment, like nothing's ever going to be perfect because obviously like, there might be some left in the test tube or whatever. But you can see from Streptococcus, this is a virus. I'm sorry, it's a bacteria. We have 29.8 and 31.6. That's pretty much the same number. 20.5, 18. That's pretty much the same number. Again, if you consider that there is going to be a little bit of error every time you do an experiment because human beings are doing the experiment, therefore, human beings make mistakes. For yeast, 31 and 32, A and T are the same. G and C, 18 and 17, pretty much the same. Here we have 27.8, 27.5, 22.2, 22.6. So again, this is actual data from Chargaff's experiments where we can see that A and T are the same and G and C are the same. Now, keep in mind, when I say the same, in science, there is always a little bit of error. When I say the same, they have to be like close enough within reason. So 2931, that's 2 apart. That's more or less the same. And again, I don't know what his margin of error was because I don't have that in front of me, but I imagine it was pretty, it was probably plus or minus 5%. All right, so let's try and do this problem here together. If a species has 35% adenine, what is the percentage of the other 3 bases? Help me out. If I have 35% adenine, what would my thiamine be?
29.
If I have 35% adenine, what would my thiamine be? Go back to the rule. What is the rule between A and T? They have to be what?
35.
It's going to be 35 because they have to be the same. A and T are the same. All right, so now I need to figure out my C and G, and to do this, I'm going to need a little bit of math. So first things first, I'm going to add up my A and T. A is 35, T is 35. So my A plus my T is 70, right? 35 plus 35 is 70. And then we're talking percentages, whenever you're talking about a percent, your percent is always it means out of 100. So that means 100 minus 70. I have 30 left over. And that 30 has to get split between my G and my C. So this 30 is going to get split between G and C, and it has to be split equally. So 30 divided by 2, G is going to be 15, C is also going to be 15. All right, any issues or questions, ladies and gentlemen? Because if not, that is where we are going to end our notes today. All right, hearing none, as always, ladies and gentlemen, you can ask questions in class, but I'm going to go ahead and stop the video.